Hey there guys, I'm your today's host, Starkwalk. Today, I'll be using this time to talk about a few fun facts about genetics. Can you roll your tongue? If you can, the chances are you will find someone else in your family who can do it too. 1 in 4 of us are tongue rollers. It is just one of those thousands of characteristics we may inherit from our parents through our genes. But do you know that every child is like their parents in some ways? But each of us has our own unique combination of genes, except for identical twins. No two people have the same genes. Well, let's take an example. Identical twins look alike because they have exactly the same set of genes. They are formed from the same fertilized egg, which splits in the mother's womb into 2 cells, each of which grows into a baby. So this leads up a question. Where are genes? Your genetic material is found in every cell in your body. Each cell has the same set up to 100,000 separate genes. Well, these genes added up to a complete set of instructions for building you from scratch, your genome. Genes are in the nucleus or control center of a cell. They are arranged along long, thin thread-like structures called chromosomes, rather like beads on a necklace. Chromosomes come in pairs, each of which carries genes for the same characteristics. So what are the different chromosomes? Different animals and plants have different numbers of chromosomes. Most snakes have 36, horses have 63, while some ferns have 500 or more. Apart from that, humans have a total of 46 chromosomes, 23 pairs in every cell. The only exceptions are sperm and egg cells, which have 23 unpaired chromosomes. When a sperm fertilizes an egg cell, each parent contributes 23 unpaired chromosomes. So the new baby has its own unique set of 46 chromosomes. So now the question is, how genes exactly work? Genes are made of a chemical called a deoxyribonucleic acid, or DNA. DNA works by telling the cell how to make the proteins that keep the cell alive and growing. Every cell in your body contains at least 10,000 different kinds of protein. Small sections of the numerous DNA molecule carry the instructions for making individual proteins. The machinery for making these proteins is on structures called ribosomes, which are outside the nucleus. The DNA itself never leaves the nucleus. Instead, small sections are copied on to another chemical called a messenger RNA. This carries the instructions for making a particular protein to the ribosomes. So what exactly are copying genes? Every time new cell is made, the DNA is copied so that each new cell has its own identical copy of all the genes. The structure of DNA makes the genes easy to copy. The DNA is copied so that each new cell has its own identical copy of all the genes. The structure of DNA makes the genes easy to copy. A DNA strand separates down the middle and spare chemical building blocks, joining up with their watching pairs on the separated strands. So one strand of DNA makes two identical new strands. Now let's take a further look of what's the problems that genes have caused. Genes can change if mistakes are made during the copying process, or if they are damaged by chemicals or radiation. They also change when they are messed up during sexual reproduction. A permanent change to a piece of DNA is called mutation. Most mutations are harmful, but a few produce helpful characteristics. Such mutations allow a species to change and evolve over time. Some combinations of genes can cause disorders and diseases. For example, about 8% of men are colorblind. They cannot distinguish the colors red and green. It is rare for women to be colorblind, but many women are carriers. This means they can pass the problem genes on to their children. As the result, scientifically, nearly a third of a human being's genes are the same as the genes of a lactus. So what does genes and environments do with each other? The final appearance and behavior of an individual is determined both by its genes and by outside influence. These influences include things like how much food it eats, what climate it lives in, and whether it has suffered disease or injured during its development. But only those characters controlled by its genes can be inherited. Now we're heading to discovery in DNA. One of the most important biological discoveries of the 20th century was made in 1953 when the British scientist Francis Crick and the American James Watson together worked out on the double helix structure of DNA. They used the information for X-ray photographs of crystals of DNA taken by scientists Morris Watkins and Rosalind Franklin to work out the DNA shape. DNA is shaped rather like a twisted letter. The shape is called a double helix. The rungs of the letter are made up of 4 chemical building blocks: adenine, guanine, thymine, and cytosine. Like the letters of an alphabet, these building blocks can be arranged in different ways to make it up the words of the genes. So genetic instructions are in the form of a chemical code. Gregor Mendel, a Czech monk called Gregor Mendel, made the first important discoveries in genetics in 1875. Mendel was experimenting with pea plants. In one experiment, he bred together plants that had white flowers with ones that had purple flowers. Instead of getting some white and some purple flowers, he found that all the new plants had purple flowers. To explain that, we have to remember that chromosomes come in pairs. Each plants have 2 genes for flower color. What Mendel showed was than the gene for purple flowers. Let's take it for instance. A in the diagram is dominant over the gene for white flowers. So if a plant has one purple flower gene and one white flower gene, the plant has purple flowers. The white flower gene is called recessive. It only shows if a plant has two white flower genes. Let's find out about our genes. The Human Genome Project is an international research program that has recently worked out the complete sequence of all the genes on our chromosomes. As yet, scientists do not know what all the different genes do. But finding faulty genes is becoming easier, and now treatments for genetic disease may result. Many medicines, such as insulin, are now produced in a large amount thanks to advances in our knowledge of genetics. Genetic engineers insert human genes, such as the gene for insulin, into harmless microbes. As they grow, the microbes produce large quantities of high-quality insulins. Genetic engineers can also insert human genes into large animals. For example, sheep have been genetically modified so that they produce medicines in their milk. Or they may perhaps have it on the other animals too. As the picture provided below, this scientist is injecting an egg cell with a DNA as a part of the process of producing a baby animal from just one of the mother's cells. This process is called cloning. Scientists also have the power to change the genes in any living thing and introduce the genes from one species into another. That's all for today's show. Please comment, like, and subscribe as always, and we'll stay tuned.